Good morning and a pleasant day to each and everyone. I am Datumolok and I am going to discuss a research article which revolves around concepts of chemiosmosis. This research article is entitled Energy Conservation by a Hydrogenase-Dependent Chemiosmotic Mechanism in an Ancient Metabolic Pathway. This is originally authored by Marie Charles Maricha in Volko Molitor and was edited by Caroline S. Howard of the University of Washington, Seattle, Washington, and was approved on February 12, 2019. Before we proceed on the discussion of this article, let us first review how chemiosmosis works. The final stage of cellular respiration or the oxidative phosphorylation, which yield the most ADP outputs, is divided into the electron transport chain and chemiosmosis. The electron transport chain is basically the shuffling of electrons from complexes 1 to 4 of the inner mitochondrial membrane, which generates the formation of an electrochemical gradient. As more and more hydrogen ions accumulate in the intermembrane space, the ADP synthase facilitates the return of the protons into the side of the membrane with a lower concentration of ions, in this case, the mitochondrial matrix. The ADP synthase takes advantage of this proton pump to build ATP molecules. This is simply known as chemiosmosis. Now let's start with the abstract of this research article. This study investigates how acetogenic bacteria, or those bacteria that can produce an organic molecule called acetate from inorganic compounds such as carbon dioxide and carbon monoxide, are able to do so using only an energy converting ion translocating hydrogenase known as the H complex. This is pretty much interesting because it employs the ancient reductive pathway of acetyl-CoA. Although the genes that encode the ECH are widely distributed in nature, the proposed function of H as an ion translocating chemiosmotic coupling site has neither been demonstrated in bacteria, nor has it been demonstrated that it can be the only energetic coupling sites in microorganisms that depend on a chemiosmotic mechanism for energy conservation. By studying how the H complex functions in the case of thermoanaerobic cuvoi, an acetogenic bacterium, it has been revealed that it can indeed serve as the simplest form of respiratory enzyme. Experiments with resting cells prepared from thermoanaerobacter cuvoi cultures grown on carbon monoxide revealed the coupling of carbon monoxide oxidation and hydrogen gas formation and the generation of a transmembrane electrochemical ion gradient. Inverted membrane vesicles, or IMVs, have been prepared for carbon monoxide grown cells, and it also produced hydrogen gas and ATP from carbon monoxide, or a chemical reductant. Finally, it became known that H activity led to the translocation of both hydrogen ions and sodium ions across the membrane of the inverted membrane vesicles. As we get to the following slides, we will also learn how this study exposed the combination of the carbon dioxide fixation with the synthesis of ATP in an ancient pathway. It is well established that the first organism to propagate in the primordial world was a chemolithid autotrophic thermophilic anaerobe. These ancient cells employ the reductive acetyl-CoA pathway. Acetyl-CoA pathway is also called Wood-Chandal pathway in dedication to its discoverers. This pathway comprehensively encompasses carbon dioxide fixation to acetyl-CoA, carbon dioxide assimilation into biomass, and energy conservation. Acetyl-CoA pathway has been closely observed with three strictly anaerobic organisms. These are the methanogenic archaea, sulfate-reducing bacteria, and acetogenic bacteria. Now, focusing on the acetogenic bacteria, these bacteria can grow heterotrophically on sugars, alcohols, and aldehydes, but they can also grow chemolithotrophically on hydrogen plus carbon dioxide or on carbon monoxide. This is possible through the reduction of two molecules of carbon dioxide to acetate with electrons derived from hydrogen gas according to this given balance equation below. Although the net yield of ADP by substrate level phosphorylation or SLP is zero, additional ATP synthesis must occur by a chemiosmotic mechanism since the bacteria do grow under lithotrophic conditions. How the acetyl-CoA pathway or Wood-Chandal pathway can be coupled to the synthesis of ATP has been solved only recently through the study of Acetobacterium woodii. This bacterium grows optimally at 30 degrees Celsius and has the simple respiratory chain consisting of a ferredoxin oxidizing, not reducing respiratory enzyme encoded by the RNF genes and an ATP synthase that are connected by a sodium ion circuit across the cytoplasmic membrane. RNF genes are not present in every acetogen, and thus a second mode of energy conservation must exist in acetogenic bacteria. Interestingly, inspections of genome sequences lead to the description of genes encoding energy-converting, possibly ion-translocating membrane-bound hydrogenases, the H complex, as we are talking about, and RNF-free acetogens, leading to the postulate that in these acetogens, RNF is replaced by the H complex. Now we have the results of this study. Two separate samples of Thermoanaerobacter tivoi were grown on glucose-containing medium and carbon monoxide-containing medium to test whether carbon monoxide oxidation is coupled to the formation of hydrogen gas in ATP. The glucose-grown cells produced only little hydrogen gas, whereas the carbon monoxide-grown cells produced hydrogen gas at a rate of 584 plus minus 3 nanomole per minute per milligram protein. This result gives a positive indication that the ability to oxidize carbon monoxide was induced during growth on carbon monoxide-containing medium. Concomitant with hydrogen gas formation, the intracellular ATP content surprisingly increased as well. It is worth noting that it is also hypothesized that the formation of hydrogen gas from carbon monoxide in Thermoanaerobacter tivoi is coupled to the generation of a transmembrane electrochemical ion gradient. This study proves this to be true. This slide provides the graph of the observed hydrogen gas formation. I have mentioned that inverted membrane vesicles or IMVs were used in the study, but how did they get to know that it is provided with an accurate result? The answer is that one-time washed IMVs were used for all measurements unless indicated otherwise to ensure the soluble proteins, particularly soluble hydrogenases, were removed from the vesicles. One-time indication from the preparation is that as hydrogen was produced, the absence of H at the membrane was ascertained. These experiments demonstrated a rather stable anchorage of the hydrogen gas evolving H complex and an only loose attachment of the CODH to the membrane. The activity of the H complex leads to the establishment of a chemiosmotic gradient that is composed of both hydrogen ions and sodium ions. It is important to test the coupling of H activity to the generation of an electrical potential across the vesicle membrane. And so this was done by incubating washed IMVs in a safe buffer and the presence of the potassium ionophore, valinomycin, and potassium chloride that can disrupt electrical fields. Each activity was stimulated by 43% with a hydrogen gas evolution rate from carbon monoxide of 139.9 plus minus 9.8 compared with 96.8 plus minus 6.5 nanomole per minute per milligram protein in a solvent control assay. This indeed demonstrated respiratory control. The sodium ionophore EDH 2120 stimulated by 30%, whereas the protonophore TCS had no significant stimulatory effect. Hydrogen gas evolution rates for the assay contained sodium ionophore, protonophore, and no ionophore were able to score 92.5 plus minus 3.3, 74.0 plus minus 3.8, or 71.5 plus minus 2.6 nanomole per minute per milligram protein. This is not in agreement with the experiment described above for whole cells. The reason remains unclear, but it may result from a weaker energetic coupling in IMVs compared with whole cells. In addition, in whole cells, acetate formation is inhibited by the protonophore, and thus more hydrogen gas can be formed from carbon monoxide. The experiments with uncoupling agents clearly demonstrate that also on a subcellular level, each activity is coupled to ion translocation and subsequent generation of an electrochemical ion gradient. This diagram includes the illustrations of all important results of the study. Now for the discussion of the study. The occurrence of two or more clusters encoding group 4 hydrogenases within an organism might be common, as it applies also to the other H-containing acetogens such as Morelia thermoacetica, Thermoacetogenium phaeum, several archaea, and even E. coli. The derived protein complexes have three modules: the electron input module, an electron output module comprising the hydrogenase, and a membrane integral domain for ion translocation and anchorage. The whole lot of experiments with whole cells in INVs of the bacterium Thermococcus anorinus revealed that protons are extruded first and sodium ions second by action of sodium ion hydrogen ion antiport mechanism. The secondary sodium ion gradient then drives ATP synthesis via the ATP synthase on the inner membrane termed as sodium ion A sub 1 A sub 0. This idea is supported by the recent high-resolution structure of the 14 subunit MBH that is obtained from Pyrococcus furiosus. The membrane arm can be divided into a hydrogen ion and a sodium ion translocation unit. The latter resembles an MRT-type sodium ion hydrogen ion antiporter. Similarly, H activity in Thermoanaerobacter coli also established a sodium ion and hydrogen ion gradient. A simultaneous transport of both ions could, for example, be explained by a promiscuous enzyme as described by the ATP synthase in methanosarcina acetiboron. A simultaneous action of one hydrogen ion and one sodium ion specific H complex, H1 and H2, or a sodium ion and hydrogen ion antiports. Although MRP genes encoding the sodium ion hydrogen ion antiport and Pyrococcus furiosus are missing in Thermoanaerobacter coli, the membrane integral subunit of both H complexes share highest sequence similarities with sodium ion hydrogen ion antiporters. There are no other apparent chain products identified as potential sodium ion hydrogen ion antiporters. Neither in the genome of Thermoanaerobacter coli nor in the genome of the closely related acetogen Methanococcus thermoacetica. Intriguingly, the existence of an electrochemical sodium ion hydrogen ion antiport has been demonstrated in Methanococcus thermoacetica. This diagram illustrates the genes that are responsible for the encoding of the H and MBH clusters. In conclusion, this work demonstrated that an H hydrogenase is a respiratory enzyme in a bacterium. A respiratory hydrogenase of an ATP synthase are sufficient to enable microbial life at the thermodynamic limit of life, and there is a second mode of energy conservation in acetogenic bacteria. This link that is provided here in blue contains the description of the experimental procedures for cultivation of the organism, preparation and experiments with resting cells, determination of hydrogen gas, carbon monoxide in acid concentration, preparation of IMVs, measurement of H activity at IMVs, measurement of the sodium isotope or hydrogen ion translocation, and determination of relative transcription level for this study. And that is all from our report. Thank you very much and have a great day ahead.